we would love it if everyone would engage with us on Twitter, on Facebook. If you hear a really interesting quote, or you want to engage more with our speakers, please hashtag Trainee Blitz and tag us research on WH on Twitter or on Facebook. The WHR cluster, I guess, is how it's spelled. So please tweet away as the presentations happen. So let's get started. And just a quick note about asking questions. So the format is each presenter will have 5 minutes, and then we usually take about 3 minutes for questions, but we don't have a huge group today. So please just unmute yourself, or you can raise your hand or type your questions in the chat box, and I'll be doing some moderating. But yeah, so those 3 minutes go quite quickly. So make sure you get your questions up, and then if there's any time at the end, we'll stick around for questions as well. So I will stop sharing my screen. And Victoria, I will get you to set yours up, and I'll just introduce you as well. So Victoria graduated from the Mount Royal University in 2020 with a Bachelor of Science in Nursing. Sorry, screen here. She then obtained her registered nurse license. Currently, Victoria is a first-year Master's of Science student with Dr. Sophia Ahmed's lab at the University of Calgary. So thank you so much for joining us, Victoria, and I'll let you take it away.
Awesome. Can everyone hear me? Okay, perfect.
Yes.
Thank you so much for the introduction and for having me. So my project is looking at the quality of life of patients initiating incremental or conventional hemodialysis and looking at it from a sex and gender lens. So just to provide some background, your kidneys are an important organ. They filter and excrete out extra fluid and toxins from the body. However, when you have kidney failure, the kidneys are no longer able to remove the fluid and toxins, so they build up in the body. Kidney failure is an irreversible and life-threatening condition, and more than 40,000 Canadians actually live with kidney failure and require dialysis. So one type of dialysis is hemodialysis. This is essentially when you are connected to a machine, and the machine is used to filter and remove the extra toxins and fluid from the blood, and it takes on the role of the kidneys. So patients, when they start dialysis, are started on what we call conventional hemodialysis. So it is done in a hospital or clinic setting 3 times a week for 4 hours. So it's quite the time commitment for these patients. And one patient-reported outcome is quality of life. And it is well evident that patients on hemodialysis have a worse quality of life than the general population. But more specifically, females on hemodialysis have a worse quality of life than males on hemodialysis. So quality of life is measured using the Kidney Disease Quality of Life Questionnaire. It is a standardized and reliable tool. This questionnaire specifically consists of 36 questions. It looks at symptoms, burden of kidney disease, as well as the impact of kidney disease on the patient's daily living. I will be using this questionnaire in my study, and it will be administered at baseline and at 3 months. So there are potential sex and gender differences that might be impacting quality of life for this population. So sex is your biological sex at birth, so male, female, or intersex. But when referring to gender, we're referring to the socially constructed gender roles and behaviors, as well as gender norms. And in research, gender is analyzed using a standardized gender score, and it looks at an individual's roles and responsibilities in their daily life. So one tool is the Genesis Proxy Gender Questionnaire. It was developed in 2015, and it's a tool that creates a composite gender score. And the questionnaire itself encompasses all aspects of gender. So it includes gender identity, gender roles, gender relations, and it also includes other factors such as socio-demographic factors. So as I mentioned, it creates a composite gender score, where a higher gender score is indicative of having higher roles and responsibilities in the household or to caretaking, where a lower gender score is on the opposite end, where you have less roles and responsibilities to your household. So one potential sex difference that could impact quality of life is the fact that females have a lower volume of water distribution. This typically indicates that females are underdialyzed and might require more dialysis in order to adequately remove the fluids and toxins. So having that extra toxins and fluid in their system can actually create or lower their quality of life. When looking at gender, it has been found that men on dialysis have more social support. So they're more often married and more often have other family or other people living at home that can help support them while on dialysis compared to women. So because quality of life is greatly impaired in the dialysis population, incremental hemodialysis is a new approach that is being initiated, and it aims to improve the overall quality of life for dialysis patients. So patients are started on a lower dialysis dose that is based on their residual kidney function when they start, but they will be started on anything less than 3 times a week. So they could start on once a week or twice a week. It's dependent on their residual kidney function. And the aim of incremental is to help transition patients onto dialysis and make it like an easier transition and also reduce treatment burden, and then in turn improve quality of life. So quality of life is a patient-identified research priority, and it is evidently lower in patients on hemodialysis, but also more specifically for females on hemodialysis. We want to improve quality of life for these patients, and incremental has the potential to do so, as it focuses on individualizing dialysis dose that's adjusted based on a patient's dynamic need. So my first objective is to determine the association between type of hemodialysis and quality of life stratified by sex. And I anticipate that female sex will be associated with a lower quality of life in both types of hemodialysis. As females are typically underdialyzed, when they're on incremental, they'll be receiving less dialysis than they would on conventional. And when looking at it from a gender lens, my objective is to determine the gender score or determine the association between gender score and quality of life by the type of hemodialysis. And I anticipate that a higher gender score will be associated with a lower quality of life in both types of hemodialysis. So having more responsibility to the household and to like other caretaking responsibilities, being on dialysis will take away from that time and create stress and therefore negatively impact their quality of life. So I just wanted to thank my amazing lab for all of their support, and thank you all for listening and for joining, and I will take any questions.
That was really interesting. I think the crossover, especially around both sex and gender in your work, where you're talking about the amount of like biological like water that is in the body, and then also the caregiving roles is really interesting. So let's see, is there any questions at all? I can see, let's see the chat. Here, everyone's just saying how great it is.
Yeah.
Let me see, let me think of a question. So with these findings, are there any kinds of interventions to improve? Like, would the goal be to roll out this incremental dialysis more to combat both those at-home responsibilities and like biological characteristics? Is that sort of the goal of this research?
Yeah, and so incremental hemodialysis is being initiated in like all across Alberta, and the aim is to have like less time on dialysis, because patients on hemodialysis have like heavily reported that the time commitment it takes away from whether they're working or going to school or have a family that they need to take care of. So by being on once a week dialysis or twice a week, like that saves them so much time instead of like having 12 hours a week designated to just dialysis. And like it is done in a hospital or a clinic, so you have to like factor in the travel time and all that stuff too. So that is the goal, yes.
Very, very cool. And then it would also address the biological like difference as well.
Yeah, so because it accounts for the residual kidney function, the dialysis dose is like, it'll basically top up what their kidneys aren't able to do, just to fully clear all the toxins from their system. But yeah, so that's why it's being initiated in Alberta.
Very cool. Well, I don't know if it's being done in BC, but I hope that's something that spreads across Canada. That's great. Any other questions at all? You can just unmute yourself and go ahead if you do. And if not, give me one more second. Okay. Thanks so much, Victoria. That was awesome.
Thank you.
So next up, we're going to have Chantelle. So if you wouldn't mind sharing your screen, Chantelle, and I'll introduce you. So Chantal is born and raised Calgarian, and after receiving a Bachelor of Science in Medical Biochemistry from UBC in 2016, she returned to Calgary to complete a Master of Science in Cardiovascular and Respiratory Sciences in 2019. Her work was nominated for a University of Calgary Graduate Medal and was Western Association of Graduate School WAG's Distinguished Master's Thesis Award in STEM. Chantal continued her studies and began a PhD in the fall of 2020 at the University of Calgary with Dr. Sophia Ahmed, assessing the cardiovascular health of transgender individuals, particularly with respect to the use of gender-affirming hormone therapy and its route of administration, duration of exposure, and frequency of use. In her spare time, Chantal enjoys doing anything outdoors and is an avid downhill skier, mountain biker, hiker, and fly fisher. All right, take it away.
Can everyone see my screen okay? Awesome, great. Thanks, Alex, for that nice introduction. And like Alex mentioned, my name is Chantal. I'm a PhD student in my first year at the University of Calgary with Dr. Sophia Ahmed. And my thesis is entitled The Cardiovascular Implications of Gender-Affirming Hormone Therapy in Transgender Women. So as a little bit of a disclaimer, I have no conflicts of interest. I personally use she, her pronouns. And I just like to recognize my white cisgender privilege and choose to do transgender research to show my support for the LGBTQ+ community. So what does it mean to be transgender? The word transgender is an umbrella term for people whose gender identity or expression differs from the sex they were assigned at birth. So a gender identity is our internal sense of gender, and alternatively, our gender expression is our public or outward-facing expression of gender. So transgender individuals whose gender identity and or expression does not align with their sex at birth can undergo a transition process to help align their physical bodies with something that represents them better. And the process of a transition can occur in basically three main ways, that being first through a social transition with a name change or different clothing being worn or coming out, as we all colloquially know the term. Kind of the next steps of a transition or a medical transition through the use of gender-affirming hormone therapy. And finally, a surgical transition, so things like breast construction. So a transgender individual can choose kind of one of three or any of all of these processes to transition, depending on their goals or their desired outcomes. However, a medical and surgical transition require the presence of a primary care physician, as they need to be kind of agreed upon, or there needs to be things that are checked off kind of for individuals to receive surgery or hormones. So specifically, my research looks at gender-affirming hormone therapy. And what is gender-affirming hormone therapy? It's the use of exogenous hormones to align the physical body with the gender identity. And that can kind of exist on any sort of continuum depending on an individual's goals. Basically, at the very far side of it, the suppression of undesired secondary sexual characteristics, all the way to the development of desired secondary sexual characteristics. And again, individuals can exist on a continuum where, depending on their goals or their point of their transition, they can move up and down this scale. And my research specifically focuses on transgender women. So the feminization process using gender-affirming hormone therapies allows transgender individuals or transgender women to reach their desired levels of feminization. And that process can look like one of many different ways, but the kind of main hallmark ways for the feminization process to occur is through the use of exogenous estrogen, exogenous anti-androgens to decrease testosterone levels, and progesterone. And kind of basically the whole point of the feminization process is to decrease endogenous testosterone while increasing endogenous estrogen. And some changes that are noted during the feminization process are things like breast development, decreased muscle mass, and voice softening. Now, with hormones, as we know in a cisgender population, there can be risks and benefits associated with their use, and there's also the same in a transgender population. So there has been quite limited data to suggest that the way in which gender-affirming hormone therapy is taken, whether that be oral or non-oral prescriptions, the dose at which it's prescribed, as well as the frequency of exposure, can be risky from a cardiovascular perspective. However, it's really unclear if these risks are due to the way, the route, the dose, or the frequency that these hormones are taken, or the resulting levels of estrogen specifically in transgender women. So like I mentioned before, the feminization process is basically to decrease testosterone and increase estrogen. But what transgender women's testosterone levels are supposed to look like and estrogen levels are supposed to look like is based on a cisgender female or a physiological female. And that's kind of unfortunate because we're subjecting individuals to maybe reference ranges that aren't great for their biology. So it kind of begs the question, you know, there are risks associated with the way in which these hormones are taken, the dose that they're taken, and the frequency that they're taken, but is it also a result of the levels that the hormones are in circulation that are kind of a product of this root frequency or dose of the hormones? So that led me to my PhD research, where I'm looking at the cardiovascular implications of gender-affirming hormone therapy in transgender women. And I want to understand the association between serum estrogen levels and cardiovascular risk, regardless of the route it's taken, frequency, dose, as well as determine the risk, the incidence of adverse cardiovascular events and risks using electronic medical records, as well as assess markers of future cardiovascular risk in a transgender population. So all of that together has quite a lot of significance in that the transgender population is a continuously underrepresented and underserved community in healthcare and research. Currently, 80% of transgender adults want to be on hormones, yet only 40% are currently taking them. And there's also the evidence that is out there is considered either lacking or low quality, and much of it compares transgender individuals to cisgender individuals. So I've just started my PhD in September this year, so we're actually actively recruiting participants for our study. Any kind of individuals who are taking hormones, whether that be birth control, gender affirming, or hormone replacement therapy. It does have to be in Calgary. People have to come into the lab, unfortunately, but if anyone is interested in knowing more information, I'd be happy to chat afterwards. A big thanks to my lab and to Alex and the Women's Health Research Cluster for hosting this, and I'm happy to take any questions.
Awesome work. This is so interesting. Wow, like, yeah, this is really, really great work. I'll just open it up and see if anyone has any questions.
I see Jennifer has one, I think. Yeah.
If you can unmute yourself, go ahead.
Awesome.
Oh, sorry.
Hi, Chantal. Great presentation. I just wanted to ask, I'm very curious about your future cardiovascular risk assessments. Do you mind sharing a little bit about that?
So we're going to do pulse wave velocity, which is a validated measure of future adverse heart events or heart risk, cardiovascular risk, and also using arterial stiffness through aortic augmentation index. So using basically probes on different parts of the body to measure the responsiveness of the vascular system. So using those as kind of predictors. Yeah. Thanks for your question.
Very cool. And you're also looking at bloods with that?
I'm looking at what, sorry?
Bloods.
Like
taking blood measures?
Yeah, we're going to take blood on our participants. I'm not sure the exact concoction of tests that we're going to run on them, but it'll most likely be kind of like a hormone panel and then a lipids panel and maybe a hepatic panel, since a lot of the hormones can have hepatic effects as well. So stay tuned.
Hi, can I be heard? Can you hear me?
Yeah.
Hi, I just had a question. It might be a little bit out there. It's Michelle McIntosh speaking.
Hi.
Sounds like a great study. I'm just thinking about cardiovascular risk and its sort of social construction due to androcentric science, and thinking about women presenting to hospital, cis women presenting to hospital, and not having, well, you know all about the symptomology of myocardial infarctions and how that really wasn't mapped onto female bodies in a biological way. So although you're looking at potential adversity with respect to hormones, I'm just putting out there also the risk that cis women experience by not having their symptoms taken seriously, like the cardiovascular risks that they have physically aren't identified, intervened upon, or rehabilitated because of androcentric bias. So it'd be interesting to see how this plays out in the future.
Mm-hmm. Yeah, and that's a really good point. You're right. Like heart attacks in women can present differently. And I think I've read a statistic that if a woman presents to the emergency room with heart attack-like symptoms, they're told that they have like anxiety and then they get sent home. So this definitely is something I think that, you know, an underserved community in research and health is transgender individuals, but definitely women of all biologies as well. So
I guess the question would be, do trans women experience the same bias?
yeah.
Yeah. So
yeah, they definitely do experience the same bias. And in addition to that, they also experience discrimination that leads them to maybe not come into an emergency room or go to their primary care physician or disclose their transgender status. And, you know, if an individual comes into the emergency room and they don't feel comfortable disclosing that they're on hormones or that they are a transgender individual, that's also super risky for their treatment from that point forward. If, you know, if they're treated for a heart attack and the physician doesn't know that they're on estrogen or testosterone, like that's a big issue too. So there's lots of ways in which I feel like we can improve the recognition of the transgender community.
Yes, good work.
Thank you.
Okay, one last question, just from a really good question from Kate in the chat here. Great presentation, thank you. Can you talk a little bit about how you think about doing respectful research with the transgender people from your position as an outsider researcher?
Yes, that's a very good question, actually. And I think, you know, a lot of me as a cisgender individual, you know, I don't want to be some cis savior and tell a story that isn't mine and be seen in a way that like I'm doing research to help someone who really can't help themselves or for whatever reason. But I choose to know and, you know, to go to sleep at night knowing that I am actively trying to improve the lives of other people, and potentially, you know, the way in which I can do that is my expertise in research. And maybe individuals who don't have that expertise in research, you know, they can improve the lives of others in different ways. But how I choose to see that is really advocating and standing in allyship with underrepresented communities, not to be telling a story that's not mine and not to be a savior of any sort, but just to be a voice in the research community for individuals who deserve the research and recognition.
That's wonderful. Thank you. Okay. All right. We'll have to move on, although I'm sure we could discuss these things for the whole hour. But Antonio, I'll get you to share your screen there. Okay. So, and Antonio is joining us from Barcelona today. So thank you for staying up late for us. So Antonio obtained his psychology degree in 2014 and a master's degree in psychopharmacology in 2015. From the University of Almería in Spain. From 2016 to 2017, he participated in the creation and ran the animal surgery unit at the Institute of Neuroscience, Autonomous University of Barcelona. Since beginning of 2017, he is a PhD student under the supervision of Dr. Riehl-Tonn. His research interests are sex differences in the TAC2 pathway when modulating normal and pathological fear memory consolidation in mice. These studies are combined with in vivo calcium imaging free moving mice. I am also interested, or he is also interested in the mechanisms by which sex hormones influence the formation of new fear memories and sex-specific mechanisms supporting fear and stress-related pathology in women and men. So go ahead, Antonio. You're on mute. Antonio, you're on mute.
Can you hear me now?
Yeah. Maybe
okay, I'm sorry.
No,
I would like to present part of my PhD project today. And first, I would like to start stating that fear is a generally adaptive state of apprehension that begins rapidly and dissipates quickly once the threat is removed, in contrast with anxiety that is elicited by less specific and less predictable threats, as said by Michael Davis, which is an expert in the field. Fear-related disorders are what we call have a common component, that is the dysregulation of fear circuitry in the brain. And these disorders include anxiety disorders, compulsive disorder, and trauma and stress-related disorders, as for example, PTSD. It's very surprising that mental diseases do not distribute equally between sexes, with men having more prevalence of drug abuse or alcohol abuse, and women having more like prevalence of stress and fear-related disorders, PTSD or panic disorders. So it's very surprising for us as neuroscientists that there's only one paper that includes includes females in comparison to 5 papers that are exclusively done in male mice. So there are different areas in the brain that do have a special role in this fear memory processing, as for example, the prefrontal cortex or the hippocampal formation, but I'm going to focus on the amygdala. The TAC2 gene is a gene that synthesizes the neurokinin B peptide. And this peptide binds to the neurokinin 3 receptor, NK3 receptor. What we do in the laboratory, we do a fear conditioning task in which we pair a tone with a food shock for 5 times in rodents, in mice in this case, and we test for fear recall 24 hours later in different contexts, exposing the animals to the same tone and measuring the freezing response to the tone. We have an opportunity there after fear acquisition. There is the consolidation window of these fear memories that we can take advantage of by giving pharmacological treatments. So what we found here is that when we gave this NK3 receptor antagonist to males after fear acquisition, 24 hours later, males recalled much lower than controls when given the drug. But very surprisingly for us, when we did the same experiments in female mice, we had a sex opposite effect. I mean, females that received the drug had a greater freezing values during the fear expression test. And not just that, we divided the females by the estrous cycle that we were during the fear conditioning and receiving the drug, and we found that during proestrus, that is when female mice present high levels of estradiol and progesterone, it's the only stage of the estrous cycle in mice when the drug is effective modulating fear memory consolidation. Also, because we know that the NK3 receptor is expressed in the gonads, we found that there is also a sex opposite regulation of sex hormones. Males tend to decrease testosterone, while females present increased estradiol in response to the drug. Also, we tested P28 animals with 29 days old, and we found no effect of these drugs. These animals have not reached yet sexual maturity, and we can assume that sex hormones have a very important role in mediating these effects. Also, we cannulated these animals and we gave the drug into the amygdala to make sure that the effect of the drug was due to a modulation of the NK3 receptors in the amygdala. And we found in female mice what we see here, that there's an increase of fear memory consolidation as measured by the freezing rates 24 hours after conditioning. There's also an approach in which we can silence these NK3 TAC2 neurons, which is called Rytz, inhibitory receptors in the amygdala, and we silence a small portion of the central amygdala, which is called the centromedial amygdala, and we found similar effects than when the drug is given intraperitoneally, systemically. So I think we managed to find the population of neurons that are behind these effects. We also identified the PI3K pathway as the main pathway that controls this memory consolidation effect, and also we measured the protein synthesis of the AKT targets, as for example CREB or GSK3-beta or mTOR, and we found a sex-opposite effect in the regulation of the AKT GSK3-beta beta-catenin pathway. So I think this is very interesting. Just to sum up all the findings, we can revert the effect of osanetan in males by giving an AKT activator to compensate this AKT downregulation, and also an androgen receptor agonist. And we can also revert the effects of osanetan increasing fear memory consolidation in female mice by giving an anti-AKT and estrogen receptor antagonist. So for us, it's very surprising that a drug has such opposite effects in memory consolidation, and we think that maybe more research should be done investigating pharmacological treatment for mental disorders. Just quickly say thanks to my supervisor, Raul Landero, and to my great team that supports me every day, Eric Velasco, Patricia Molina, Laura, and Nacho. And thank you very much for having me today. I'll be happy to answer your questions.
Great. Thank you so much, Antonio. That's really interesting work.
Thank you.
All right, I'll open up the floor for questions.
I have a couple questions.
Yes.
One of which I'm just wondering, the drug itself, does it have any other effects on the mouse behavior that you've been able to find in your research, or that you've seen in other research that people have done? Just wondering what
Well,
other effects it has on behavior.
yeah, thank you for your question. We looked at different behaviors, as for example, anxiety and locomotive behavior, and the drug is totally safe because it doesn't show any effect on locomotive behavior or anxiety. Also, this drug is safe for humans. Actually, there's been a couple of studies with humans using this drug for another psychopathologies, as for example, schizophrenia, and it's safe, well tolerated, but it's not useful for schizophrenia. So maybe, I don't know, we could give another use to this drug.
Awesome, thanks. And then another question I just had was, I was wondering what age the mice were that you were using, and were they adults, and if you think that the same effects would occur in, say, an adolescent stage group, or what do you think of that?
Yeah, we actually use 7-8 week old mice that are young adults, and we found with the P28, P29 experiment that mice need to be sexually mature to have the effect of the drug. So I don't know, we don't know if it's because they need high levels of sex hormones, or maybe it's just a developmental effect of maturation, but they need to be after puberty to have this effect on memory consolidation.
Amazing, thank you so much.
Thank you, Travis.
I just have a quick question. I'm thinking about the memory. Actually, not a question, but I wonder if you're familiar with the research. I can't think of the researcher's name. They're in Texas, the university, and they were looking at mice and memory. And I'm thinking about the question about age of mice. They put mice through a maze. They were looking at the brain-uterus connection in mice. And its effect on memory. And so they put mice through a maze, and the mice could remember the route through the maze until they removed their uteruses. And then the mice had no memory. And so I wonder if you know, sex hormones, but also the communication between the brain and the uterus. You've talked about estrus as a factor. I wonder if you've thought about it.
Yes, I've done some research about it, and we know that the removal of the ovaries, for example, that reduces estradiol levels, have a very strong impact on memory consolidation, memory in mice. For example, in Alzheimer's disease, it increases the risk of beta-amyloid plaques in the brain. And also, if estradiol is not present, female mice tend to have lower rates of memory consolidation, and they have disrupted fear processes, like they show a lot of fear and they are unable to learn the extinction of fear. So they remain like afraid all the time the tone appears. Every time the tone appears, they cannot learn that this is safe when the shock is gone.
Fascinating.
Thank you very much.
Anyone else? I have a quick question. You mentioned that the drug had been used in schizophrenia, and you said that there may be other uses. I know more research has to be done, but do you have any hypotheses about what other uses it would?
Yes, for example, in men, for example, not in women, in this case, we could use this drug after trauma, after an accident, after a traumatic event, during this consolidation window, to somehow reduce the effects that these trauma may have in the future, and reducing the risks of developing phobias or post-traumatic stress disorder. Also, this drug could also enhance memory, but not just fear memories. It could also enhance other type of memories that we are working now on that, like recognition memory, and it could be also useful for those patients that have memory deficits, as for example, in dementia or other mental disorders that have memory issues.
Very cool.
Thank you.
Thank you so much. That's really great work.
Thank you very much.
Thanks. So Jennifer, you're up next. So I'll get you to share your screen and I'll introduce you here. So Jennifer Williams is a PhD candidate in the Department of Kinesiology at McMaster University. Her present research focuses on the short and long-term impact of hormonal contraceptives on cardiovascular and metabolic indicators in females. And she has recently pivoted her research to look at the underlying mechanisms of contraceptive hormones in vascular and endothelial cells. Committed to the advancement of women in STEM, Jennifer volunteers with the McMaster Women in Science and Engineering group and is a graduate trainee with the CIHR Institute of Gender and Health, and is involved in starting up a Sex and Gender in Health research network at McMaster. She's excited to share her current PhD research progress and hear of other women's health research from the WHRC trainees. So welcome, Jennifer, and go ahead.
Fantastic, Alice. Can you hear me okay?
Yeah.
Awesome, thank you. So hi, everyone. My name's Jennifer. I'm a PhD candidate from McMaster University. Thanks so much for that introduction. I'm excited to be sharing some of my recent research with a pivot that we've been doing during this pandemic, looking at oral contraceptive hormones in endothelial cells. So to provide a little bit of background, oral contraceptive use is quite prevalent here in Canada. Approximately 16% of Canadian women 15 to 49 years old use oral contraceptive pills, representing about 1.3 million women across Canada. However, about 4 in 5 women have used contraceptives at some point in their lifetime. So it's quite prevalent. But how contraceptive use impacts cardiovascular health, in particular in examining those early risk factors for cardiovascular disease, is relatively understudied. So I'm going to show you a cross-section of an artery here. Our lab looks at the function of major conduit arteries in the body, for example, the brachial artery in the arm. And so to orient you to the slide, the innermost layer of conduit arteries, there are endothelial cells that you see there in pink, and then smooth muscle that surrounds the endothelial cells. Now, these endothelial cells change and respond to blood flow as well as the production of vasodilators that diffuse into surrounding smooth muscle around the endothelial cells and subsequently cause vasodilation in the arteries. Now, endothelial cell and smooth muscle cell function are integral for vascular health, and so dysfunction is associated with the progression of cardiovascular disease through atherosclerotic mechanisms. So we're really interested in looking at how endothelial cells function. And so typically, so this is a cross-section of what we would see typically in our lab looking through ultrasound technology. So the last time I presented here at the Trainee Network, I was presenting on some human research that we were doing. So we were doing work in the lab looking at, through ultrasounds to look at the vascular tissue within individuals. We were also taking blood to look at various biomarkers, and we were also doing exercise performance testing in these women to look at cardiorespiratory responses to hormonal contraceptives. We were about midway through doing the study and had to do a pivot. So I've now been doing some pretty cool cell research. I'm not a wet lab researcher, but I'd like to say I'm becoming one in shifting from my dry lab work. So I've spent almost a year to the date shifting my research, pivoting to cell work, and bringing my undergraduate lab students along for the ride. So you can see here me. I have a harness Zoom call to show my undergraduate students what I'm doing while we experience these restrictions here in Ontario. So what are we looking at in cells? So when we're looking in cells, we're interested in looking at the role of contraceptive hormones on endothelial cell mechanisms. And what I mean by that is endothelial cells and their function are largely dependent on the production of a potent vasodilator nitric oxide, or NO, that you see on the screen. This is produced by a key enzyme called endothelial nitric oxide synthase. So that's what produces the nitric oxide. And those are acted upon by contraceptive hormones such as ethinylestradiol, which is the main synthetic estrogen found in contraceptives, as well as progestins. There's several of them found in contraceptives. They interact with various receptors that are on the surface of endothelial cells to either up or down regulate enos and thus nitric oxide. So that's what we're interested in studying. And the literature around this is quite sparse. There's only been a couple studies, none of which have looked at the different contraceptives that are currently found in more recent modifications of oral contraceptives. So that's what my research is focusing on. So really the purpose of my study is to comprehensively examine the effects of contraceptive hormones on vascular endothelial nitric oxide pathway. And the significance of this is it's going to hopefully fill some really important gaps in our understanding of female vascular physiology and the influence of exogenous sex hormones. So in terms of methods, what we're doing right now, we're midway through our cell research presently. What we're doing is we're exposing cells, particularly human umbilical vein endothelial cells, or HUVECs for short, to 24 hours of hormone exposure at physiologically relevant levels. And that's really important for being able to draw some comparisons between human and cellular work. So in designing our experiments, what we've done is we expose cells in these cute 6-well plates, we put them in, and then we expose them for 24 hours, and we look at a variety of different outcomes. So in particular, we look at different receptors quantities, we look at endothelial nitric oxide synthase, both the inactive and active versions, and then we also look at the production of nitric oxide. So I have some cool photos. We've been doing Western blots to take a look at protein content, and we've also been using fluorescent probes that you see on the screen there to look at the amount of nitric oxide as well as intracellular oxidative stress. So cell ROS is one of our probes that we use to take a look at how much fluorescence there is baseline and then in response to a physiological stressor, acetylcholine. So that's what I've been up to in terms of the work that we're currently doing. And I hope within the next year to be able to come back to the Trainee Network and share our results, because we are seeing some pretty cool things coming out of that research. So I'd like to thank you for listening, and thanks to my team from the Vascular Dynamics Lab here, including my supervisor, Dr. Maureen McDonald. This was the last group photo that we took together here as a lab group. So now it looks a little bit more like this nowadays, but I'm grateful for their support in helping me to pivot my research. So I'm happy to take any questions.
Awesome, super interesting work, and pretty amazing to be able to pivot in that amount of time. I have to say that. And I also love the little Zoom camera thing. That's so cute. Okay, does anyone have any questions?
Hey, Jennifer, Chantal.
Hi.
Hey, you were saying the cell ROS that you're measuring. What exact, is it just like superoxide levels, or are you measuring other things as well?
Yeah, so it's a superoxide anion probe, and so it's not assessing, it's assessing total reactive oxygen species that are found within the cell. There is another probe that we can look at for mitochondrial specific oxidative stress, but we are looking at just overall oxidative stress within the cell.
Nice, cool.
Yeah, great question.
I have a question. It's a little bit of a selfish one, but
That's okay.
so what's the age range? Because you said oral contraceptives, so it would be women of reproductive years, I would guess.
Yeah, so in our human trial, we're looking at premenopausal women who are using oral contraceptives a variety of years. We've seen individuals anywhere from freshly on, 2 months on, all the way up to like 12 years, I think is our oldest participant on contraceptives. And we look at a variety of different measures in terms of our human work, but when translating it to the cell models, we've had to scope a little bit more closely to looking at the mechanistic pieces that are involved. But yeah, we're hopeful that it'll provide some really important insights into our human work when we return.
For sure. Any plans to transition this into like a menopausal group, like with hormone replacement therapy? Because I mean, the nitrous oxide pathway would be really interesting for cardiovascular for sure.
Absolutely, yeah. So in terms of our human research, we've been mainly focusing on the premenopausal just because there are some research questions stemming from existing projects that we've been working on. So we're working on that, but definitely we've been doing some collaborative research projects involving postmenopause. I'm particularly interested in, there's some techniques that we've been refining to take serum from humans and expose to cells so that we can look at the interactions between the human and the cellular interface. And in that, we're hoping to look at some bloods that we have collected for postmenopausal women and any collaborations that folks are interested in. So hopefully that might be a plug to this network. We might be in chatting.
Yeah, I mean, I'd love to follow your work personally. So, but I can
Awesome.
chat with you later.
Sounds good.
Any other questions?
I just have a question.
Sure.
In terms of the amounts that you're doing, I mean, some women are taking only progesterone and not combination pills. So I wonder if your research would inform the impact on endothelial with the absence of estrogen exposure. And also I wonder, women who are taking more hormone, the women who are taking oral contraceptives to suppress periods are getting extra. So there's been not a lot of research to inform the benefits and risks of that. I wonder if your work will help to inform that as well.
That is a fantastic question. I'm really glad that he brought up the progestin only piece, because there actually is an absence of literature. There is no literature on the effects of progestin only pills on women. There are a couple studies that have looked at IUD use, which is particularly interesting. But I'm glad that he brought that up, because I have an undergraduate thesis student, and her research right now is focusing on progestin only mechanics within cells. And we're expanding this summer. Hopefully, we've just heard that we're going to be heading back in person to looking at how emergency contraceptive pills, so a supra-physiological dose of progestins, how that's affecting both humans, so doing an acute study on that, as well as we've just finished up our experiments looking at that in cells. Really cool data coming from that work, so hopefully we'll get to share that pretty soon. But yes, I think that's a really important question and definitely warrants investigation, and it's on our radar. But thanks for sharing that.
Especially since they're reviving the rods, the progesterone-only rods, and also lots of research done on medroxyprogesterone. So I'll look forward to your finding.
Sounds good. Yeah, the use of long-acting contraceptives are definitely on the rise. They've doubled in the last couple of years, so last 5 years. So that would be definitely an important thing to look at. Thanks for sharing.
Okay. Any other questions? No. Okay, sounds good. Thanks, Jennifer. Yeah, we definitely will have to have you back, because I'd love to see those research results.
Sounds good. Thanks so much, Alex.
All right. Last but not least, Lara, if you wouldn't mind sharing your screen, and I'll introduce you. Last and certainly not least. All right. Okay, so Lara is currently completing her honors Bachelor of Science and Behavioral Neuroscience at UBC, and is excited to be conducting her undergraduate thesis at the Swell Lab. She's interested in fields intersecting psychology, neuroscience, endocrinology, and women's health. She aspires to investigate historically understudied topics in psychiatric and medical research, such as sex differences and sexuality, birth control, pregnancy, postpartum, periods, nutrition, eating disorders, and hormone imbalance. Awesome. So go ahead, Lara.
Hi, everyone. So I'm Lara. I'm an undergraduate student at UBC, and I'm working with Dr. Samantha Dawson this year at the Swell Lab. And I've been looking into a project of body image self-consciousness during sex and sexual function problems in pregnant couples. So sexual health is a fundamental component of well-being, and problems with it can lead to adverse health outcomes, higher rates of relationship dissolution. And sexual functioning here is described as having difficulty moving through stages of desire, arousal, and orgasm, and not being satisfied with the outcome with the frequency and outcome of sexual behavior. So pregnancy is a vulnerable time for sexual function in both partners, interestingly, with up to 90% of pregnant people experiencing clinically significant problems with sexual function, and this percentage is about 30 to 40% in partners. So because of this, it's really therapeutically useful to look into any linked factors that could help us find interventions. And one such factor is body image, which is how we evaluate ourselves, how we think about our bodies, and body image changes throughout pregnancy. And it's also poor body image is a risk factor for sexual function problems outside of pregnancy, both in men, women, clinical and non-clinical populations. So because of this, we thought that poor body image may be particularly linked to sexual function problems in pregnancy, considering all the physiological changes going on, changes in types of sexual activity that are possible, and probable increased self-consciousness in the individual. So not a lot of research has looked into this at all. That has had very varied results and has focused primarily on the pregnant individual, on just the facet of sexual satisfaction, and has only used general body image measures. And these facts kind of ignore the interpersonal nature of sexuality, as well as other aspects of sexual function, such as arousal or desire, and also neglect a more measure of body image that is more related to sexual activity. So because of this, we sampled both couple members. We comprehensively assessed sexual function and all of its subscales, and used a measure of body image related to partner interactions and sexual activity. Our question was how an individual's body image self-consciousness links with their own and their partner's sexual function during pregnancy. And we hypothesized that one's own higher body image self-consciousness would link with one's own lower sexual function and also link with the partner's poor sexual function as well. So data came from a larger longitudinal study of first-time parent couples during their transition to parenthood. And we used data from a survey at 20 weeks pregnant that assessed sexual function and body image self-consciousness, among other things. Our total sample was 62 couples, and the average sample, the average couple was heterosexual, Caucasian, highly educated, married, and had an annual income of over $80,000. We also did include 9.7% of our sample was non-heterosexual couples, and 79.5% and 17.8% of pregnant individuals and partners had clinically significant problems with their sexual function, which about matches the literature. So these are the two measures we used, the body image self-consciousness scale. And then for sexual function, individuals chose between the FSFI and the IIEF. I won't read these out, but an example of an item on the BISC is during sexual activity, I am concerned about how my body looks to my partner. And an example of a sexual function desire item is over the past 4 weeks, how often have you felt sexual desire and how would you rate your level of sexual desire? So to analyze our data, we use the actor-partner interdependence model, which is very useful for allowing us to look at actor effects. So how your own self-consciousness affects your own sexual function or links with your own sexual function. And then also partner effects, which is how your partner's self-consciousness links with your sexual function. And interestingly, we did not find any significant actor effects. Nor did we find a significant partner effect that meant that pregnant individuals that had partners that were higher in self-consciousness didn't tend to have any additional problems with arousal or desire or anything like that. But we did find a significant partner effect going the other way, where higher body image self-consciousness in the pregnant individual was linked with lower sexual function of their partner. And we think that this may be due to higher body image self-consciousness in pregnant individuals, making them less comfortable performing certain types of sexual activity, and which may make it more difficult for the partner to become sexually aroused and therefore lower their level of sexual function. And this really highlights the interpersonal nature of sexuality. Still, we did not see any actor effects, which we were expecting based on previous literature, and hypothesized that this could be due to the fact that the average relationship length in our sample was about 6 years. They were having a child together. They were probably very comfortable with each other and secure in their long-term relationships. And because of this, they had lower body image self-consciousness, which was shown in our data. And then body image self-consciousness was not as associated with sexual function in this sample as it was in other studies that used younger individuals, probably in more formative years of their relationships. So overall, we think that other factors may be a little more relevant to sexual function problems in pregnancy particularly, such as physical discomfort and nausea in pregnant individuals, and that exploring body image self-consciousness in postpartum may be relevant since of all the social stigma and pressure put on these individuals to lose weight. And this could then be useful for understanding more persistent sexual function problems. So thank you all for listening. I want to thank my lab and Dr. Dawson and welcome any questions or suggestions you may have. Thanks.
Thank you. That was great. Really, really important work. All right, let's see if anyone has any questions.
Hi, Laura, I have a question for you. Did you look into how many babies a person was having? Like twins, triplets, just a single baby?
So
Because I wonder if that would like exacerbate feelings of of discomfort and stuff anyways.
So luckily the study that was looking into all these factors, because a lot of different studies come from this data, we had screenings where it had to be a singleton pregnancy and it had to be the first child, and there couldn't be any other children living in the household at the time either, because it was looking at the sort of transition.
Cool. Do you think it would change if they had more than, if they were not a singleton pregnancy?
I feel like there wouldn't be any reason not to expect significant like similarities that I could personally think of. Maybe some sort of exacerbation with considering that having like twins and stuff like that, that's automatically like can lead to more changes in physiology and having more trouble during pregnancy and something like that. But from what I can tell, I think results would be quite similar even if we'd included them.
Cool, great research.
Thank you.
Did you ask any questions about consent? I mean, we know violence really increases during pregnancy. It can really increase in pregnancy. It can also go away during pregnancy. But I'm just thinking if you're looking at sexual function and pregnancy, it might be important to know whether or not there's presence of violence in the relationship and if all acts of sex are consensual.
That would be very important to know as well. I am not aware of any questions that were looking into that, because I'm thinking I've sort of gone over everything I can think of. I think we sort of, yeah, I guess we sort of assumed that relationships were, I don't know, like healthy, normal relationships, considering the like length, again, that not that that means anything, but they were like sort of signing up for the study, the length of time they've been together, the fact that they were interested and willing to sort of have answer these super personal questions about their lives and their relationship and stuff.
Yeah, and you may be right. You may be right.
But yes, for sure. It would be useful, I think, to look into that, or to at least have one question that sort of hinted at that in some way.
Yeah, it even hooks into the question about numbers of children. I mean, you're talking about how many fetuses people are carrying at a time, but if you've got someone who's young and has had multiple children, that can sometimes be a red flag in terms of non-consent or control, in terms of contraception and reproductive control in the relationship.
Mm-hmm, mm-hmm. Yeah, so we did, because of that, we did recruit and ask for it to be, well, not just because of that, but yeah, we did control in a way where I think the average age was around 30, 32. They were all first time parents.
Sounds like great research, especially undergrad. Very impressive.
Thank you.
That's great. Yeah. And it's a place to start, right? So, and then, maybe those questionnaires didn't capture any data around interpersonal violence, but, as this research grows, those could be other directions. So, but great work. Any other questions? No, in the chat, just saying great presentation. That's awesome. Really interesting work. Really, really interesting work. And I'll echo the last comment, especially at an undergraduate level. That's amazing. All right, we're just slightly after 1 o'clock, so I think we'll wrap it up here. Thank you so much, everyone, for coming out this afternoon, whether it was over your lunch hour or making time for it in the day. Really great research presentations from everyone. I'll just do a short plug that if you can either come again and present again, or if you know of anyone who would like to participate in these sessions, we try and do once a quarter. So and a lot of the way we recruit for it is just word of mouth. So if there's other people in the lab, or there's participants who know of somebody, maybe I'll just drop my email in the chat here. But you can really reach out to anybody in the cluster and they'll kind of direct you in the right way. There we go. But yeah, feel free to email me as well. And with that, I think I'll end it. So thank you so much, everyone.